Hello, I'm Avery. I'm at Cornell University, and in the second part of this talk, I'm going to tell you a little bit about some of our research studies trying to understand the molecular basis for the allergic response in mast cells. So here's a summary of how the allergic response occurs. We have an allergen that generates an IgE response. That IgE then coats mast cells and basophils so that the second time you get exposed to an allergen, mast cells degranulate and you get these symptoms of an allergic response. So we're very interested in trying to understand the molecular basis for this degranulation response because we like to understand how to better target this process to prevent both degranulation and the development of these symptoms. So, as I told you in the first part of my talk, blocking mast cells and blocking mast cell degranulation can reduce the release of these components. And by reducing the release of these components, we actually reduce the symptoms. But what is the molecular basis for this? As I told you, the drug cromolyn is able to do this, but we don't quite understand how cromolyn works, and also cromolyn has side effects. So what we do know is that the mast cell has a receptor for IgE, and that receptor for IgE is actually very similar to receptors that are found on T cells, the T cell receptor, or on B cells, the B cell receptor. And all those receptors, what they have in common are cytoplasmic tails that have what are called I-time motifs. And these I-time motifs interact with families of kinases that lead to an intracellular signaling pathway that eventually leads to degranulation and histamine release, as I discussed in part 1, as well as cytokine production. And so what we're very interested in is trying to understand how these pathways actually lead to these different effects. So what we do know is that the Fc receptor for IgE triggers an increase in intracellular calcium. And that increase in intracellular calcium is very important for both degranulation and cytokine production. And one of the compounds that has been discovered that can actually inhibit this calcium release is a molecule that was first reported by Avery labs as an immunosuppressant. And that molecule, which I'll name BTP for short, is actually very interesting because it actually can inhibit calcium signaling in cells. And so we were very interested in trying to understand whether this particular compound would also inhibit the mast cell response in vivo. So to do that, what we did is we took mice and we injected them with IgE that was specific to a molecule called DNPBSA. And when we injected that IgE will now circulate within the mouse, will coat the mast cells and basophils, and now when we inject the mouse with the allergen, within 3 minutes, we can determine whether the mast cells will release histamine, which we can then analyze. We also determined whether BTP would block this effect. And so what we found was that when we injected mice with BTP an hour before we gave them the allergen, we could get reduced histamine release, indicating that BTP actually blocks the release of histamine from these mast cells. The other thing we wanted to do is determine whether this mast cell effect would lead to differences in temperature. One of the things that happens when you expose mice to IgE and the allergen is they actually lose body temperature. And we can measure that body temperature by measuring the temperature over time in the presence or absence of the BTP. And so in that experiment, what we did, we injected the mice with IgE, and an hour before we give them the allergen, we gave them BTP, and then we measured the amount of the temperature changes that occurred. What you can see here is a difference in body temperature in these mice. And you can see that the mice that received the vehicle actually lost body temperature, whereas the mice that received BTP did not lose body temperature. And so what that experiment indicated to us was that BTP was able to inhibit both histamine release in mast cells, but also the effects of mast cell degranulation, that is, temperature loss. So BTP then can inhibit mast cell degranulation and can inhibit the release of the mast cell granule contents. So we wanted to find out whether BTP could affect mast cells directly. And so fortunately, we can generate mast cells very simply by taking bone marrow from mice, culturing them in the presence of cytokines, interleukin-3, and stem cell factor. And 4 to 6 weeks later, we have a population that's largely mast cells, almost 100% mast cells. We can then take those mast cells and analyze them. And the way we did this is we took the mast cells, we incubated them with IgE overnight by arming them, and then we can add the antigen, and over time we can analyze these mast cells. And so we did that, and what we found was that in mast cells that were just treated with vehicle, we got nice degranulation, whereas mast cells that were only treated with BTP and triggered with IgE, we no longer got degranulation, indicating that BTP actually inhibits the degranulation of mast cells, blocking them from releasing their contents, including histamine, heparin, and TNF. So the next thing we wanted to find out is what is the target of BTP? And so we collaborated with a couple of chemists, Blake Peterson and his student Laurie Mottram, and they synthesized for us the BTP coupled to a linker in biotin. And what we could then do is take that BTP biotin linkage and couple it to a bead and determine, using standard biochemical techniques, which proteins would actually interact with the BTP moiety. And so that experiment is shown over here. You can see that we would take control beads and coat them with just the linker in biotin, or beads that had the BTP. We would pass a protein extract over those beads. We would wash away all the extraneous material, and then we would analyze the proteins that actually specifically interacted with the beads on SDS-PH gels by mass spectrometry to identify BTP-associated proteins. And so we did that experiment using lysates from cells, and what we found was that a prominent protein that was purified on these BTP beads was a protein called Drebrin. And so Drebrin is actually an actin-binding protein, and it actually can interact with actin and leads to the polymerization of actin when you put it in cells. We also found that when you mutate two residues within the actin-binding domain of Drebrin, we could abolish binding of BTP to Drebrin, indicating that BTP interacted directly with Drebrin and not in an indirect manner. And those residues are shown here in red. If we mutate lysine 270 and lysine 271 to methionines, we completely abolish the binding of BTP to Drebrin. Whereas if we mutated arginine to methionine or glutamine to leucine, we maintained the interaction, indicating that this interaction was specific. So we could show then that BTP interacts with this actin-binding protein Drebrin, and Drebrin is an actin-regulating protein. So that it introduced a whole new area that we could explore to try to determine what was the molecular basis for this Fc receptor response. So again, here's the pathway that I showed you, and what we showed you is that calcium, shown in red, is actually inhibited by BTP. I also just showed you that Drebrin was identified as a BTP interacting protein. And so the question was, where does Drebrin fall within this pathway that is triggered by the Fc receptor? And so we did some experiments to try to determine whether Drebrin would be able to affect the activation of a number of these different pathways. So in order to do that, we actually generated Drebrin knockout mice. And we did that by procuring the embryonic stem cells, which had a cassette that was trapped within the gene for Drebrin, which resulted in the cells not being able to make the protein. We generated mice from those embryonic stem cells, and then we examined the brains of those mice, which also expressed Drebrin, for the expression of Drebrin. And you can see here in this Western blot that the wild type's brains expressed full-length Drebrin, whereas the brains from the Drebrin deficient mice had no Drebrin at all. We then went on to further characterize these mice to determine whether their mast cells were functional. And what you can see here is skin from the either wild type or Drebrin deficient mice stained with toluene blue, and in the black arrows you can see where the mast cells are found in the skin, and you can see that mast cell development was completely fine in the absence of Drebrin in these mice. We also looked at electron micrograph images, and you can see again that the ultrastructure of these mast cells was completely fine whether Drebrin was there or not. So we were fairly confident that in the absence of Drebrin, at least in vivo, mast cells were able to develop and they were found in the right place. So the next thing we decided to do is ask whether these mast cells was functional in vivo in this analysis that I talked to you earlier. And here what we did again, we armed the mast cells in vivo with IgE by injecting IgE, and then the next day we challenged them with the allergen, and 3 minutes later we collected these mice and determined whether there was any histamine. And again, what you see here is that wild-type mice generated a lot of histamine when we gave them the allergen plus IgE, whereas the Drebrin-deficient mice had a much reduced histamine response under the same conditions. So these experiments indicated to us Drebrin was really important for the release of histamine from mast cells in vivo when we triggered them with IgE. The second experiment we did was the experiment that I talked about earlier, which is, do these Drebrin-deficient mice actually lose body temperature when we inject them with allergen? And so we did the same experiment. We armed the mast cells in vivo with IgE, we gave them the allergen, and then we monitored their temperature for up to 60 minutes later to determine whether they lose body temperature. And what you can see here is that the vehicle wild-type mice that received vehicle, they lost body temperature. In the absence of Drebrin, they no longer lost body temperature. And wild-type mice that are treated with BTP also no longer lost body temperature. So in both the knockout as well as the compound, we got the same result, that is, passive systemic anaphylaxis was actually reduced in response to allergen when we inhibit with BTP, when we knock out the gene Drebrin. So the next thing we wanted to do then is look at the individual mast cells and ask whether we saw the same result. So again, we took bone marrow from mice, in this case either wild-type or Drebrin-deficient mice. We grew them out in culture for 6 weeks, and then we coated them with IgE overnight and then triggered them with allergen to determine whether they would respond. And so again, we're looking at the activation of mast cells by the IgE receptor. And first we're going to look at calcium responses to see whether calcium was affected in the absence of Drebrin. And that experiment is shown here. We take mast cells and we load them with a calcium-sensitive dye. We then coat them with IgE and then trigger them with the antigen. And you can see that the wild-type cells, in light gray, increase calcium, and that calcium stays up for a long time. Whereas the absence of Drebrin, these mast cells increase calcium initially, but the calcium doesn't stay up. It starts to decrease over time. And in the bottom graph, you can see the slope of that calcium decay is a negative slope in the absence of Drebrin in these mast cells, whereas it's a positive slope in the wild-type cells, indicating that in the absence of Drebrin, what we're actually affecting is the prolonged calcium release that we see that's required for these mast cells to degranulate and release their contents. So, mast cell degranulation then requires the activation of Drebrin, the activation of the Fc receptor, and releases these contents, histamine, heparin, and other cytokines. And so we then asked whether we could see reduction in degranulation in the absence of Drebrin in these mast cells. So that experiment is shown over here. We can stimulate these mast cells, either wild-type mast cells or Drebrin-deficient mast cells, with IgE and the allergen, and in a concentration-dependent manner, you get an increase in degranulation as measured by the release of beta-hexosaminidase. So in wild-type mast cells, you get an increase in beta-hexosaminidase release, and when Drebrin is missing, you can see here that the mast cells degranulate much less compared to the wild type. So the absence of Drebrin then affects mast cell degranulation in vitro, in addition to reductions in intracellular calcium over time. So what about cytokines? So I told you in the first part of my talk that activation of mast cells with allergen not only releases histamines, but also releases cytokines. And those cytokines can have important effects on the immune system. So we therefore did a similar experiment where we took mast cells and stimulated them and asked whether Drebrin, or the absence of Drebrin, would affect the cytokine release. And the cytokine release, some of those cytokines are dependent on calcium, and some of the cytokines are not dependent on calcium. So we looked at two different patterns of cytokines, calcium-dependent cytokines and calcium-independent cytokines. And that experiment is shown over here. And the top two graphs are two calcium-dependent cytokines, interleukin-2 and GM-CSF. And you can see here in the filled circles, wild-type cells produce IL-2 and GM-CSF perfectly fine, but in the mast cells that are missing Drebrin, you can see that now they no longer release the calcium-dependent cytokines. By contrast, if we look at IL-6 on the bottom graph, which is not a calcium-dependent cytokine, you can see that there's very little difference between the wild-type cells and the Drebrin-deficient cells in cytokine production. So these experiments show that Drebrin plays a critical role in regulating both calcium signaling leading to degranulation, but also calcium signaling leading to cytokine production, and only those cytokines that are calcium dependent are affected by the absence of Drebrin. The other thing that we wanted to find out is where in this pathway does Drebrin lie? And so we did a number of experiments where we examined the activation of the Src, Syk, and Tec kinases, and there was no difference. We asked whether PLC gamma activation was affected by the absence of Drebrin, and there was no difference. And so what we could conclude then was that Drebrin plays a role in a parallel or different pathway that is not upstream of PLC gamma, but actually still affects calcium in a manner that we don't quite understand. Now, one of the things that I told you about was that the activation of Drebrin is important for actin polymerization. And so we wanted to find out whether Drebrin would affect actin in these cells. Now, actin is really important for the structure of the cell, but it has also been shown to be important for degranulation and the movement of granules within cells. And so the idea that Drebrin plays a role not only in regulating calcium signaling, but also in regulating the actin cytoskeleton prompted us to look at the actin in these cells. So we did some fairly simple experiments to determine that. First, we took either wild-type or Drebrin-deficient mast cells and stained them with phalloidin, which stains F or filamentous actin. And what you can see most apparent is that the wild-type cells actually have nice actin around the perimeter of the cell. But if we look at the Drebrin-deficient cells, you can see that they have more actin, and the actin is actually distributed not only in the perimeter of the cell, but starts to extend within the cell. So we wanted to quantify this a bit more. And so we did flow cytometry with these cells using labeled phalloidin. And you can see here that wild-type mast cells in solid have actually less actin, if you look at the mean fluorescence intensity, than the mast cells that don't have Drebrin. So in the absence of Drebrin, these mast cells actually have increased F-actin, which may be affecting their response. So we wanted to explore this a bit further and ask what happens to the F-actin when these cells are actually stimulated. And so what we did is we took these mast cells and stimulated in the same way that we stimulated them before with IgE and allergen. And we looked at F-actin over time in these cells. And so this is a bit of a complicated slide, and I'll take to it slowly. On the right, you can see that the wild-type mast cells start out with actin at time 0. Most of the actin is at the edge of the cell, at the perimeter of the cell. By 2 minutes after activation, you can see that the F-actin starts to move within the cell with the increase in the signal at 2 minutes, 5 minutes, and 10 minutes. And then by 30 minutes at the bottom, the cell looks fairly similar to a cell that hasn't been activated. So in wild-type cells, there's an increase in F-actin that's reorganized away from the perimeter of the cell towards the middle of the cell, and then it moves back to the perimeter of the cell. If we look at the Drebrin-deficient mast cells, you can see that at time 0, actin is distributed all over the cell, not only at the perimeter. And that pattern doesn't change much as we stimulate the cells over time. So IgE Fc receptor-induced changes is different between the wild-type cells and Drebrin-deficient cells in space and in time. And we can quantify this by comparing the difference between the Drebrin-deficient cells and the wild-type deficient cells at each time point statistically. And you can see here that on the y-axis in this, the graph on the left, that at time 0, there's a huge difference in the F-actin localization in these cells, and that changes as the cell is activated. But in all cases, there's a statistically significant difference in the location of F-actin in the wild-type cells compared to the Drebrin-deficient cells. So what these experiments indicated to us was that Drebrin was regulating the F-actin structure, or superstructure in these mast cells. So we asked the question, can we relax the F-actin superstructure in these Drebrin deficient cells and rescue the function in the absence of Drebrin? So we used a compound called Latrunculin B. What Latrunculin B does is it binds to F-actin and increases its mobility, or it reduces the amount of F-actin in cells. And what I show you here on the right is our flow cytometry graphs of Drebrin-deficient mast cells treated with Latrunculin B. And you can see here in the table on the left, as we increase the amount of Latrunculin B, we reduce the amount of F-actin in these cells. So we can use Latrunculin B to tune the amount of F-actin in these cells. And we can now ask the question, if we reduce the amount of F-actin in these Drebrin-deficient cells, can we rescue degranulation? And that experiment is shown here. So here in the filled circles are wild-type mast cells that we've stimulated with IgE and antigen in a concentration-dependent fashion. And we get an increase in degranulation over that period. In the absence of Drebrin, in the open circles, you can see that Drebrin-deficient mast cells actually degranulate much less. However, when we treat these Drebrin-deficient mast cells with either 0.5 or 1 micromole of Latrunculin B, we can partially rescue degranulation, indicating that allowing F-actin to be more fluid in these cells allows these cells to now be able to degranulate. So we can summarize then by placing Drebrin in this pathway in a way, so that the Fc receptor triggers the activation of these tyrosine kinases, which then leads to the activation of a number of different pathways, including Drebrin. And what Drebrin does is control the amount of F-actin in these cells to control either the calcium signaling and or degranulation of these cells so that they can release their contents, produce cytokines, and participate in the allergic response. And so with that, I'd like to thank the people who did the work in the lab. Most of this work was done by the people shown in red. Mankit Law was the main person who drove most of the work, with help from a number of postdocs and graduate students in the lab. We collaborated with Lori Motram and Blake Peterson, along with Tomo Shirao and Hiro Yamazaki at Gomai University.